The Purdue Research Foundation Office of Technology Commercialization moves Purdue innovations to the public. Among those innovations is a method to quickly process food samples used to detect food pathogens like Salmonella in fresh produce, meat, and other food. The technology was developed by a team led by Michael Laddish, distinguished professor in the Department of Agricultural and Biological Engineering and the Weldon School of Biomedical Engineering, and director of the Laboratory of Renewable Resources Engineering.
Food safety in the United States is very good. The detection of food pathogens, microorganisms that cause food illness, in fact, has an excellent record, although, as we all know, there are sometimes exceptions. The government agencies, the FDA, the USDA, and industry that constantly seeks to detect food pathogens can identify not only the pathogen, but the source of it, as well as the type of pathogen. And the challenge they now face is being able to do more samples and do them more quickly, so the time between when a food pathogen might occur and when it is in fact detected would be shortened.
Laddish's team has developed a method that processes food samples in hours instead of days and concentrates microorganisms to accelerate detection of pathogens. The method processes 2 samples at the same time and will be expanded to handle more simultaneously.
So the purpose of the assay in our method is to detect food pathogens, microorganisms that cause food illness. The method itself will homogenize the food. It could be a vegetable, could be chicken, it could be a fruit, and then filters out the particles with the liquid extract being processed further. The processing then removes the water, concentrates the microorganisms that are present, and delivers them in a form that is able to be interrogated by existing methods, including PCR, immunoassays, and also traditional plating techniques.
They're looking to develop the technology through commercial partners in the food safety industry and in cooperation with researchers at the FDA and the USDA.
Currently, we can process 2 samples at a time. The next step will be to increase the number of samples we can process simultaneously to 96. This will make our method compatible with existing detection methods, which are based on 96 well plates. And so as a consequence, what we're looking for is a technology development effort that will enable us to do the simultaneous samples, and also commercial partners that will help us build enough instruments that can be tested in the industry and in government laboratories.
Purdue University's patented technologies include new cancer treatments, alternative fuels, biomedical devices, agricultural innovations, and others that are available for licensing through the Office of Technology Commercialization. For more information, email innovation@prf.org.